Well, hey AP Bio, hope you're doing well when you watch this video. This is lecture 1 in Unit 6, DNA, Molecular Nature of Genes. We're going to dive into the history of DNA, the fascinating history of DNA, and also the chemical structure and specifics about it. But I don't know about you, but when I think about DNA, lots of things pop into my mind, but it's got to be Jurassic Park. Of course, the DNA molecule looks like this guy. There's DNA. And of course, that scene where they're kind of on the ride and they see the eggs and all sorts of stuff like that. It's a really good movie. But here's the big idea. Basically, the evidence that DNA and RNA carry genetic information in all life forms. Okay, so all life is connected. Began with a series of historical experiments that led to an understanding of how the structure and interaction of molecules facilitate the replication and transfer of DNA and RNA across membrane. So that's basically kind of where we're going here, historical experiments. And so there's, computer image of DNA. That's a big question, right? What is the structure of DNA? There's Watson and Crick. Watson, Crick. We'll talk about them more in a second. Before we begin, I'd pause right here to see if you can do these 4 bullet points before you begin. We already watched the Discovery of DNA video. It was very insightful and awesome. I just wanted, this is what this video is about, trying to kind of dive into a little bit more of the specifics about that. So, by the early 20th century, a chromosomal theory of inheritance had been developed. Mendel's genes were on chromosomes. Everybody started figuring this out. And so, what led scientists to believe that DNA is the genetic material? Evidence began to accumulate. The evidence first started with a man named Frederick Griffith. Frederick Griffith was working with a Streptococcus pneumoniae, causes strep throat, pneumonia, various things like that. One pathogenic and one harmless. So pathogenic, of course, causes disease, pathos, suffering. And so when he mixed heat-killed remains of the pathogenic strain, so he killed them with heat, remains, with living cells, some living cells became pathogenic, so they transformed. So he called this transformation, a change in genotype and thus phenotype due to assimilation of foreign DNA. So here's a picture from a textbook. And so basically you have living smooth cells, pathogenic control, mouse dies. Rough cells, non-pathogenic control, mouse is healthy. He killed S cells, mouse is healthy. And so here's where the brilliant part of this experiment comes into play. He took a mixture of heat-killed S cells, all right, so mouse lived, and living R cells, mouse lived. Mouse lived, mouse lived. Combined those two in the sample, injected the mouse, and the mouse dies. And so from the blood, he saw smooth cells. And so this was transformation. The R cells literally took up some of the DNA left over from the S cells and became the pathogenic strain. And so this, transformation, we see this today. Genetically modified organisms, GMOs. You go shopping for groceries or with your parents or grandparents or aunts and uncles, you will see non-GMO or GMOs. It's a very valuable tool in modern biotechnology today. People argue about, is it right or wrong? Is it safe or not safe? But genetically modified, transformation. GFP is a green fluorescent protein. A jellyfish gene codes for GFP. Jellyfish gene is isolated and transferred to a bacterium or the embryo of a plant, pig, or mouse. You can see here, and these animals are now glowing in the dark. This is kind of used as trackers to see, usually they're just not trying to make glow-in-the-dark animals for Halloween. They're also trying to do studies and various things. How do they know if the DNA they wanted to get incorporated into the genome gets incorporated? Well, they put a tracer on it, a label. And so if it glows, they put this tag this with the gene that they're studying. It's pretty amazing. And so these are called transgenic organisms, whole new genetically transferred organisms. And you can click on this and do some research about this. We'll talk more about this down the road, but it's fascinating. Just because of DNA structure and how it can be transferred between organisms and still carry the code from the original one. Also, Oswald Avery, a gentleman by the name of Oswald Avery, contributed to the evidence that DNA is the transforming element, and it carries the genes. So basically, he took Griffith's experiment. Science builds on itself, right? So took the heat-killed S cells, lipids and carbohydrates, removed the lipids and carbohydrates. And so protein, RNA, and DNA remained. All right. And so he added enzymes that broke down protein enzymes that broke down RNA, and enzymes that broke down DNA. He added R cells, and transformation occurred, even if there wasn't any RNA or protein. But when DNA was destroyed, no transformation occurred. So that is another, point of evidence that shows that DNA is where genes are located. So another experiment, some of the history here came from studies of viruses. So some viruses called bacteriophages or phages are widely used in molecular genetics research. So a virus, we're all too familiar with viruses these days, right? DNA or RNA enclosed by a protective coat. Viruses must infect cells and take over the cell's metabolic machinery. Yeah, they're hijackers. They have to hijack the cells and then use the cell's own machinery to make more viruses. So here's a bacteriophage. Kind of looks like a lunar landing module. It's pretty crazy. And here's the genetic material. This is, of course, 100 nanometer. I mean, it's like crazy, ridiculously small. And so this is just a bacteriophage. Okay, COVID doesn't look like this, by the way. And so what happens? DNA is injected, hijacks the cell's machinery. The cell actually starts making all these viruses because of the own machinery. It gets hijacked by the virus and various proteins that the virus carries in with the DNA. And so it's just pretty amazing how viruses work. And so two people studied this, Hershey and Chase. And there's a famous experiment. What they did, they designed an experiment showing that only the DNA of the T2 phage and not the protein enters an E. coli cell during infection. So there's your blank there. But they determined that was the DNA, not the protein. How did they do this? There's a picture of them. How did they do this is that they actually radioactively labeled them. And so what they did was they mixed radioactively labeled phages with bacteria, okay? And then they agitated the mixture, okay? But they did two different batches. Batch one, the phages were growing with sulfur. And so sulfur is in protein, okay? And then they agitated it, centrifuged the mixture so the bacteria formed a pellet at the bottom. And what they found was the radioactively phage protein was found in the liquid. It wasn't found in the pellet. Centrifuge is like a really fast, spinning machine, increases gravity, separate things. Phages were grown with phosphorus, which is in DNA, and they saw when they agitated the cells that the radioactively labeled material was in the pellet. Thus, the DNA became radioactively labeled, and that's what led to the infection of the cells. Okay, and then another historical scientist that I would be aware of is Erwin Chargaff. And so he found that A binds to T and C binds to G. So many different mnemonic devices to remember this, but apples in the tree, car in the garage is a very simple one. So two findings became known. The base composition of DNA varies between species, and then A binds to T, that's equal, and G binds to C. And so this is, of course, Chargaff's rule. So here's some data from several papers of his. And you can see, of course, it's not exactly the same due to mutations, but if we did statistical analysis, it is, it's right there. A binds to T, C binds to G. And so from this, you can figure out the missing pieces doing just random math. But that's some of the data that he found in his studies. And then we started getting into building the model of DNA. X-ray crystallography had a huge role in this. Positions of atoms can be inferred from the diffraction pattern of X-rays. And so here's where we get to Rosalind Franklin. Rosalind Franklin, of course, there's photograph 51. And she is a hero in, the world of biology. She studied the structure of DNA using X-rays. And remember from the video, her photo, given again without her permission, really opened the eyes of Watson and Crick to build the model correctly. So this provided evidence, these diffraction photos, that DNA had the following features. First off, it's a helix, and then some portion of the helix is repeated. And so this is kind of what happened. X-ray beam, crystalline DNA, diffraction pattern, and there's her famous photograph 51. And so chemists knew that nucleotides consist of a sugar deoxyribose, a phosphate group, and nitrogen-containing bases, purines, adenine and guanine, and pyrimidines, cytosine and thymine. And so using all this information, building upon years and years of research, also debatably taking research unethically, to be honest, because Wilkins showed photograph 51 to Watson and Crick without telling Rosalind Franklin. So not necessarily Crick's and Watson's fault, but nonetheless, Crick and Watson combined all the knowledge of DNA to determine its structure. So Franklin's X-ray crystallography convinced them it was helical. Density measures suggested there are 2 polynucleotide chains in the molecule, and so modeling showed that they must be anti-parallel, like a highway. They go in opposite directions. And so here's that picture again. And so here's DNA, 3' and 5'. So here's just a picture of DNA in a textbook. You can see how the helix going around and around and around, oxygen, hydrogen, phosphorus, lots of different elements. Watson Crick suggested that the bases are on the inside, and of course A binds to T, C binds to G, and then it's double-stranded helix of uniform diameter. So the chains are held together by hydrogen bonds, which will be a very important thing when we talk about replication and transcription. And so 2 hydrogen bonds between A and T, 3 hydrogen bonds between C and G. And so in the sugar phosphate backbone, the phosphate groups are bonded to the 5' carbon of one sugar and the 3' carbon of the next. So you can see this is numbering carbons, 1, 2, 3, 4, 5. You'll go into that in chemistry and stuff. But you can see here that 3' end and the 5' end. And so you can see that they run in opposite directions. Okay, so here's a picture from another textbook. They're anti-parallel phosphate groups attached to the 5', nitrogen space attached to the 1' carbon, and then here's the OH attached to the 3' carbon over here. 5' end is flipped, so you can see 5' and 3' go in opposite directions. Covalent bonds between sugar phosphates, super strong. Hydrogen bonds, not as strong. Van der Waals interactions stack and help them stay together. So you can see that lots of bonding, that's unit 1, lots of bonding happening in DNA. So the surfaces of A and T and C and G base pairs are chemically distinct, which is super important because DNA is there, right? But we've already learned that just because you have a code doesn't mean it's expressed, and methylation and all sorts of different things. And so they're chemically distinct, which has a huge role to play for replication and gene expression. So in closing, DNA structure is essential to its function, storage of genetic information, precise replication during cell division, mitosis, by complementary base pairing, right? We're trying to make 2 clone cells. We got to have the exact same book of DNA. Susceptibility to mutations. Yeah, mutations can become stable. They can lead to new genes possibly, or, bad mutations that can lead to problems, such as loss of function or like cystic fibrosis. Expression of the coded information as the phenotype. So there's a code, and then we build the phenotype from that. So I love reading the comics. I used to do it with my dad all the time every Sunday. Here's Calvin and Hobbes. When you look at me, it's clear that my genes contain the evolutionary perfection of earthly DNA. I am the culmination of creation. With no tail? I don't think so. Stop, Pat. My butt doesn't need aesthetic enhancement. So Calvin and Hobbes, it's a great thing. But yeah, DNA, nature versus nurture, it does tell us how to develop, but it doesn't determine how we'll develop. Of course, we can't develop a tail like, Hobbes thinks. All right, so here's your last 2 kind of questions. Given a polynucleotide sequence such as GAATTC, explain what further information you would need in order to identify which end is the 5' end. And then while trying to develop a vaccine for Streptococcus pneumoniae, Griffith was surprised to discover the phenomenon of bacterial transformation. Based on the results in the second and third panels, figure 13.3, what results was he expecting in the 4th one? And we kind of already talked about, but just in case, here's the panels right here. And they're talking about this one. Okay. And so go ahead and answer those couple of questions. But that is lecture 1, the history of DNA and its chemical structure. Hope this was helpful, and hope you and your family are doing well. God bless. Jesus loves you.